In 2005, a paleontologist dissolved part of a 68-million-year-old Tyrannosaurus rex bone. She expected dust. Instead, she found something soft, flexible, tubular, structures that looked like blood vessels. For decades, biology textbooks had taught that proteins cannot survive millions of years. Yet structures consistent with blood vessels were there, inside a dinosaur fossil. In March 2005, paleontologist Mary Schweitzer published a paper in the journal Science that would spark decades of scientific debate. Schweitzer and her team at North Carolina State University had been studying a Tyrannosaurus rex femur recovered from the Hell Creek Formation in Montana. The specimen, catalogued as MOR 1125 at the Museum of the Rockies, was approximately 68 million years old. The research team used a standard laboratory technique to study the bone's internal structure. They placed small fragments in a solution of EDTA, which dissolves the mineral component of bone while leaving organic material intact. When Schweitzer examined the demineralized fragments under a microscope, she observed structures that appeared impossible given their age. Transparent, flexible, hollow tubes that branched like blood vessels. Some measured 10 to 40 micrometers in diameter. Under scanning electron microscopy, they showed morphology consistent with microvasculature in living bone tissue. Inside some of these tubes, the team observed round microstructures that resembled red blood cells. In the bone matrix, they found what appeared to be osteocytes, bone cells still preserved within their microscopic chambers. The structures reacted positively to antibodies designed to bind to collagen and hemoglobin. Control experiments using modern bone showed similar antibody binding. Tests on bacterial biofilms showed no reaction. Schweitzer's paper documented these findings with electron microscopy images and chemical analysis. The conclusion was cautious. Soft tissue structures appeared to have survived in this fossil. The scientific response was immediate and divided. Critics argued the findings contradicted fundamental principles of organic chemistry. Even under ideal preservation conditions, kinetic models predicted complete breakdown of proteins within a few million years. Some researchers suggested the structures were mineral precipitates. Others proposed contamination. A few dismissed the findings as laboratory artifacts. Over the next 2 years, Schweitzer's team replicated the findings in 8 additional specimens. The 2007 paper in Proceedings of the Royal Society documented similar preservation in multiple Cretaceous fossils. In 2009, the team published mass spectrometry data. They had extracted proteins from an 80-million-year-old hadrosaur specimen and sequenced peptide fragments. 7 sequences matched vertebrate collagen type 1 with hydroxyproline and hydroxylysine, modifications unique to vertebrate collagen that bacteria do not produce. Independent laboratories began testing the findings. In 2011, researchers used synchrotron radiation to analyze Cretaceous vertebrate bone and detected molecular signatures consistent with preserved proteins. In 2015, electron microscopy revealed the 67-nanometer banding pattern characteristic of collagen in demineralized fossil bone. By the mid-2010s, the question had shifted from whether preservation occurred to how it was possible. In 2013, Schweitzer's team published a hypothesis in Proceedings of the Royal Society, proposing that iron chemistry could stabilize proteins across geological time. When animals die, red blood cells rupture, releasing hemoglobin. Each hemoglobin molecule contains iron atoms bound within heme groups. As tissues decay, iron is liberated. Iron reacts with oxygen and water, driving what chemists call Fenton reactions, processes that generate free radicals. Free radicals normally accelerate tissue decay by attacking proteins through oxidation. But Schweitzer's team proposed that under specific conditions, this chemistry could preserve rather than destroy. Fenton reactions produce reactive aldehydes. Aldehydes can form covalent crosslinks between protein molecules, binding them into stable networks. This is chemically similar to formaldehyde fixation used in laboratories to preserve biological tissues. The hypothesis: if crosslinking occurred rapidly after death, before bacterial enzymes degraded proteins, it might stabilize structures for extended periods. To test this, researchers extracted blood vessels from fresh ostrich bone and treated one group with iron-rich solutions. Both groups were stored at room temperature. After 2 years, the iron-treated vessels remained soft and structurally intact. The control vessels had decomposed within weeks. Chemical analysis detected carbonyl groups, molecular signatures of crosslinking, in the preserved vessels. The experiments suggested iron-mediated cross-linking could extend tissue survival beyond normal decay timescales. But 2 years is not 68 million years. In 2019, Schweitzer's team published more detailed analysis in scientific reports. They examined the original T-Rex vessel structures using synchrotron-based infrared spectroscopy. The analysis identified two preservation mechanisms. First, iron-catalyzed cross-linking via Fenton chemistry. Second, glycation non-enzymatic reactions between proteins and sugars that form advanced glycation end products. Both mechanisms create carbonyl crosslinks between protein molecules. The infrared spectra detected these signatures in the fossil vessels. The patterns matched experimentally aged collagen and tissues where glycation is accelerated. The team also identified goethite, an iron oxide mineral embedded within vessel walls. Goethite forms when iron oxidizes slowly in the presence of organic material. Its presence indicated iron had been associated with proteins since shortly after death. The chemical evidence suggested a preservation pathway. Iron released from hemoglobin catalyzed rapid protein crosslinking, protecting tissue from bacterial enzymes. Over geological time, continued glycation reinforced the protein network. Accumulation of iron oxide minerals provided additional structural support. This mechanism requires specific conditions: rapid burial to limit oxygen exposure, chemically stable burial environment with neutral pH and minimal groundwater flow. The Hell Creek Formation provided these conditions. The dinosaur was buried in fluvial sediment that became chemically reducing as organic matter consumed oxygen. The surrounding rock protected the bone from infiltrating groundwater. The iron hypothesis offered the first chemically plausible mechanism for soft tissue survival across deep time. In September 2025, physicist Hans Hallen at North Carolina State University published a study providing direct molecular evidence for hemoglobin preservation in dinosaur bone. The research, published in Proceedings of the Royal Society A, used resonance Raman spectroscopy. This technique identifies molecules by measuring how they vibrate when struck by laser light. Different molecules have unique vibrational frequencies. Resonance Raman offers additional selectivity. By tuning the laser wavelength to match a specific molecule's absorption, researchers can amplify its signal while suppressing others. Hallen's team targeted heme, the iron-containing ring structure in hemoglobin. They analyzed bone extracts from the original T-Rex femur and the hadrosaur Brachylophosaurus canadensis. For comparison, they analyzed modern ostrich bone and human blood. The Raman spectra from both dinosaur samples showed characteristic heme vibrational signatures. This confirmed that iron porphyrin complexes had survived in fossil bone. More significantly, the spectral patterns indicated the heme remained bound to protein. In functional hemoglobin, heme sits within a protein pocket formed by globin chains. These protein-heme interactions produce spectral shifts that distinguish bound heme from free heme. The dinosaur spectra showed these shifts. The patterns matched oxidized hemoglobin, where iron has been oxidized from ferrous to ferric state. This was critical evidence. Bacterial proteins do not bind heme in the same molecular geometry as vertebrate hemoglobin. The specific protein-heme configuration revealed by resonance Raman is characteristic of vertebrate globin proteins. The study provided direct evidence that fragments of hemoglobin, specifically heme groups still bound to portions of globin chains, had survived in dinosaur bone. The spectra also revealed degradation pathways. Some heme groups had lost iron atoms, leaving metal-free rings. Others showed oxidative damage. Importantly, the data showed associations between heme and goethite, suggesting physical linkage between degrading hemoglobin and precipitating iron minerals. This supported the proposed preservation mechanism. As hemoglobin breaks down, released iron forms minerals that coat and infiltrate the protein matrix, providing structural reinforcement even as proteins degrade. 2 months earlier, in January 2025, researchers at the University of Regina studied specimen RSKM P25238 Scotty, the largest T-Rex ever discovered. They analyzed a fractured rib using synchrotron X-ray tomography at the Canadian Light Source. The scans revealed three-dimensional networks of mineralized structures within the rib. These structures ranged from 100 to 500 micrometers in diameter. They branched and interconnected across bone zones, running perpendicular to the bone's grain. The morphology matched blood vessels involved in bone healing. The rib showed evidence of an incompletely healed fracture. The preserved structures appeared consistent with angiogenesis, new blood vessel growth during healing. Chemical analysis detected elevated iron in the structures, identified as goethite and hematite. The structures also showed calcium, phosphorus, sulfur, carbon, and nitrogen. The team characterized these as mineralized casts, iron oxide precipitates that had infilled and replaced original vascular structures. The study did not recover extractable soft tissue, but the mineralized architecture preserved the three-dimensional geometry of the original vascular network. This was methodologically significant. Previous studies required demineralization, which destroys spatial context. The synchrotron study preserved that context, showing vessel-like structures in their original anatomical positions within intact bone. Together, the resonance Raman and synchrotron studies provided convergent evidence. Both confirmed molecular or structural remnants of blood vessels had survived, stabilized by iron-mediated preservation. The 20 years following Schweitzer's 2005 publication illustrate how scientific consensus develops through evidence accumulation and methodological refinement. Initial skepticism was methodologically appropriate. The preservation of soft tissue across 68 million years was extraordinary given prevailing decay models. Early criticism focused on contamination. Replication attempts produced mixed results. Some groups recovered similar structures, others did not. The resolution came through understanding preservation conditions. Molecular preservation depends on specific burial environments. Specimens from oxidizing conditions or those that experienced groundwater infiltration typically show no preservation. Specimens from reducing environments with stable chemistry are more likely to contain preserved material. The second wave of criticism targeted analytical methods. Critics argued immunological tests could produce false positives and questioned mass spectrometry database matching. Laboratories addressed these concerns through methodological improvements. Studies incorporated multiple antibody controls and refined mass spectrometry protocols. The 2009 collagen sequencing paper was particularly important. The identification of hydroxyproline and hydroxylysine provided chemical specificity beyond simple sequence matching. Theoretical criticism came from chemistry. Calculations based on known hydrolysis kinetics suggested no collagen should remain after 1 million years. This prompted experimental work on preservation mechanisms. The 2013 and 2019 studies demonstrated that cross-linked proteins resist hydrolysis far longer than uncross-linked proteins. Laboratory experiments lasting 2 years established proof of principle that iron treatment dramatically slows decay. The 2025 resonance Raman study addressed remaining identification concerns. Unlike immunological or mass spectrometry methods, Raman spectroscopy provides direct structural information based on molecular vibrations. The spectral data permitted no alternative interpretation. The detected structure was consistent only with protein-bound heme characteristic of vertebrate hemoglobin. By 2025, evidence from multiple independent laboratories using diverse techniques had established working consensus. Molecular remnants of soft tissues can survive in fossil bone under specific conditions. This consensus is reflected in publication venues. Papers have appeared in leading journals including Science, Proceedings of the Royal Society B, Scientific Reports, and Proceedings of the Royal Society A, all peer-reviewed by experts in paleontology, biochemistry, and materials science. The findings have been presented at major conferences and incorporated into university curricula. The National Science Foundation has supported research through competitive grants. Debate continues regarding specific interpretations and quantitative survival limits. This ongoing research activity characterizes healthy scientific discourse. The case illustrates how science resolves disagreements through evidence accumulation, methodological improvement, and mechanism elucidation. The process took 2 decades, involved dozens of groups, and required integration of multiple analytical approaches. In March 2005, examination of bone fragments from a 68 million-year-old Tyrannosaurus rex revealed structures morphologically consistent with blood vessels. 20 years of subsequent research has established that molecular remnants of soft tissues can survive in fossil bone under specific geological conditions. The preservation mechanism involves iron released from hemoglobin catalyzing cross-linking reactions that stabilize proteins. Combined with iron oxide mineral precipitation, this creates protein-mineral hybrids resistant to decay. The mechanism operates when burial rapidly seals carcasses from oxygen and when groundwater chemistry remains stable. These conditions are not common, but occur with sufficient frequency for systematic investigation. Chemical analysis has confirmed collagen peptides through mass spectrometry. Spectroscopic methods have detected preserved protein signatures. Resonance Raman spectroscopy has demonstrated that hemoglobin fragments persist in fossil specimens. Synchrotron imaging has visualized mineralized vessel structures in 3 dimensions within intact bone, confirming anatomically appropriate positions consistent with original vascular architecture. These findings have methodological implications. Proteins provide sequence data for phylogenetic analysis. Isotopic compositions constrain reconstructions of ancient physiology. Vessel morphology informs metabolic and growth estimates. Progress required integration of paleontology, biochemistry, analytical chemistry, and materials science. Techniques from medical research and forensics found application in studying ancient fossils. The trajectory from controversial discovery to working consensus illustrates scientific evaluation of unexpected findings. Initial skepticism prompted replication, methodological refinement, and theoretical development. Evidence accumulated through systematic investigation. Mechanisms underwent experimental testing. Independent laboratories confirmed results. Current understanding is based on available evidence and existing capabilities. Future developments may reveal additional complexity or require model modifications. This provisional nature is inherent in scientific methodology. The story also reflects persistent investigation despite skepticism. The molecular preservation hypothesis contradicted established models and faced significant criticism. Overcoming this required 2 decades of experimental work and innovation. Research continues. Studies are expanding to additional taxa and geological periods. New techniques are extracting information from increasingly degraded materials. Models are being refined to predict preservation in different environments. The recognition that molecular information can survive fossilization has changed perspectives on what can be learned from ancient remains. Fossils are increasingly recognized as chemical archives that may preserve traces of biochemistry and physiology across deep time.